Hello everybody, welcome to the start of our new chapter. This is going to be chapter number 21, which is going to explore the world of our immune system by taking a look at how it has an innate as well as an adaptive way of defending our body against any type of foreign pathogen. Let's go ahead and let's get started. Now, when you hear the immune system, I think many of us will think along the way of a defense system, something that's going to protect us from a bacterial or a viral infection. And along the ways, that's actually exactly what it does. But it turns out that it's going to be a very complex, very detail-oriented system. And it starts off by the fact that your immune system is roughly divided into two defenses. The first defense is what we like to call our innate defense system. And this is a very non-specific type of defense, meaning that all of us have the same level of innate defenses. These are going to be things that are considered our first and our second line of defense. So for instance, the skin will be your first line of defense because that's the first thing that a bacteria cell would encounter as it tries to penetrate into your body. A second line of defense would be your typical white blood cell, fever, or inflammation. These are all lines of defense that are very general. All of us are able to generate them, and they pretty much work the same for all of us. And then you have a system, a defense system, that's going to be very specific. It's going to be tailored based on what you are exposed to. And that defense system is going to be called the adaptive or specific defense system. And this one is going to work by developing very specific T and B cells. And these T and B cells will be able to identify very particular antigens, foreign receptors, and they will also be able to generate a memory of their attack. So it will allow us to build up immunity for certain reactants. So innate is basically what everybody has, and then the adaptive will vary from individuals depending on what you expose yourself to and depending on what particular T and B cells are being generated. So take a look at this very simplified diagram, and it basically just shows you that if we're looking at innate defenses, it's the same for everyone. So it's going to be things like our first line, which is the surface barrier, which is like your skin and your mucous membranes that you would find in your orifices. So like the mouth, the nasal, the anal, the reproductive cavities, those have lots of mucus in order to trap any type of pathogen that tries to enter into the body. You can also, under innate defense, have your second line of defense, and these are going to be your everyday white blood cells and some other defenses. Like, for instance, we could take a look at inflammation. We can take a look at proteins that are going to be specific for antimicrobial detection, the development of a fever, so we spike our internal temperature. And on top right here, we can see our phagocytosis. Those are going to be our cells that are able to bulk eat. They're going to be very important. And then we have these cells called natural killer cells. Now, some of you might remember the natural killer cells. They were mentioned in our chapter 17 when we discussed blood. They are a specific lymphocytes that are able to identify any type of cell that doesn't belong to the body, and they go ahead and attack it. But they don't do phagocytosis. They have another way of inducing apoptosis, which we'll discuss in a little bit. Either way, these phagocytes, these natural killer cells, the method of inflammation, fever induction, and the use of antimicrobial proteins, those are all seen in every single human being, so they are an example of an innate defense. Your other option is going to be based on exposure, meaning what you are exposed to as you go ahead and you live your life and you travel and you eat different foods and you go out into the world. And that is then going to be called our adaptive defense. And our adaptive defense will allow us to develop B cells, which are part of our humoral immunity, which we're going to explore in a little bit, and our T cells, which are going to be part of our cellular immunity. The B cells and the T cells, once again, chapter 18, when we talked about blood, they are specialized lymphocytes, and these T cells and B cells will become activated only when they are exposed to a foreign antigen, and they will develop a specific defense for that foreign entity. And in addition to attacking it, they will also form a memory so that if you ever become exposed to that foreign entity again, you will have a quick recall system, and a lot of times you will have less symptoms, and some people might not even fall ill upon second exposure to that same foreign entity. So let's go ahead and talk a little bit more about our innate defenses. And then as we progress through the chapter, we're going to go ahead and go into adaptive. That way we'll get a nice little overview of everything that's going on in our immune system. So when we take a look at our innate, as I said to you before, you have your first line of defense, which is going to be the skin and the mucous membrane. And then you have your second line of defense. And these are where you're going to find your particular cells, as well as fever, inflammation, and some of your proteins. So on this list right here, we just go ahead and we just highlight some of the mechanisms. So you once again see the mention of the phagocytes, and probably the most important phagocytotic cell that we have are going to be our macrophages, which come from our monocytes. We have our natural killer cells, abbreviated as NK. Those are once again going to be a type of lymphocyte. And then we have our ability to induce inflammation, which is such an important response for our body. So we will talk about the exact steps that go into that process. And then towards the end of taking a look at our innate defense system, we'll take a look at some of the proteins like interferons and complement proteins that we use to alert the system, as well as the induction of fever. Now here is a quick illustration to remind us of the process of phagocytosis. Remember, phagocytosis is an example of endocytosis, which means that you're allowing entities to enter into the cell using a vesicle, a little carrier. Phagocytosis is also informally called bulk eating because you're allowing a large material to enter into the cell. So go ahead and take a look at the picture. Let's start on the left-hand side. And what you're seeing here in purple is a good old-fashioned macrophage. And this macrophage, as you can see, is extending its cytoplasm. Now, these little extensions, these are called pseudopods, and it's due to the fact that your plasma membrane is very fluid, so it's able to stretch out. And as it stretches out into the environment, it's able to recognize and interact with the foreign entities that are drawn over here in green. These are going to be our bacteria cells. And as you can see, the pseudopods will make physical contact, and what the macrophage will do is it will do phagocytosis to immobilize the bacteria, in essence to bulk eat it, so that we can get rid of it. So come on over to the right-hand side, and you can see the steps that are going to happen during phagocytosis. So we see that the bacteria will go ahead and enter into the macrophage through the use of a little vesicle. And once it's contained within the vesicle, we do see that there will be some phagocytotic enzymes that are going to be released in combination with the lysosome organelle. And these enzymes that are basically an acidic hydrolase enzyme, meaning it's going to create an acidic environment on the inside, it's going to allow for the bacteria cell to literally pull apart and fragment into smaller pieces. And it will allow the bacteria cell to be destroyed. And what do we do with the debris of the bacteria cell? Well, we see that we're going to release it back to the outside. Now notice on number 5 how it says sometimes we have exocytosis, and that's of course when the vesicle is used to remove the debris out of the cell and it can exit. And when you see that it's exiting, we call that exocytosis. Macrophages work great if you have a small scale type of infection, but they also allow you to kind of have time to alert your more specific cells like your T cells and B cells over to the site of injury, because they're going to start attacking and immobilizing the bacteria in this case, or any type of foreign entity. So it is a really nice example of a second line of defense. Your natural killer cells, which are, as I said, specific lymphocytes or modified lymphocytes, these are also an example of our innate system. Now, your natural killer cells will basically look for any type of cell that is not identified as a self-antigen. A self-antigen is an antigen that belongs to you. It's kind of like a stamp that says property of, right? So what the natural killer cell does is it basically roams around your body and it checks the antigen on the cells it encounters. And if it sees a self-antigen, it leaves it alone. But when it comes across a cell, like maybe a virus-infected cell or a cancer cell, any cell that's abnormal, it will notice the fact that those cells will not have that self-antigen. And it will then go ahead and mount its attack. Its attack will be based on inducing apoptosis, which is programmed cell death, and it will not phagocytose. Instead, it's going to use different chemicals to go ahead and penetrate through the foreign cell and cause it to induce its cell suicide, aka apoptosis. Let's click over to the next slide so we can take a look at this in an illustrated example. Alrighty, so here we have our enemy cell. So this could be a cancer cell, this could be a virus-infected cell, and here's my natural killer cell. And what we're seeing here is that the natural killer cell has made contact with the quote-unquote enemy. It has recognized the fact that it doesn't have a self-antigen, so it doesn't belong. So the way it's going to go about killing it off, it's going to release perforins. Now, perforins are going to allow the polymerization of a little opening right over here. You can see them forming this little channel, a little opening, a little hole in the plasma membrane of the enemy cell, so the foreign cell that doesn't belong. Once the perforin have perforated, the cell membrane, we see that the natural killer cell will next release its next set of enzymes. These are going to be called granzymes. And the granzymes will enter through the perforin polymerization process inside the enemy cell and will induce the cell to go through apoptosis, cell death. And as you can see right here, notice how the cell starts to what they call bleb. It starts to become fragmented, small little pieces. And with apoptosis, the really nice thing is that the cell starts to implode, meaning it gets smaller and smaller and smaller, and eventually usually a macrophage will come by and it will phagocytose the remaining cell membrane of the enemy cell, thereby getting rid of any evidence that the cell ever existed. I would highly recommend that if you have some time, go ahead and take a look at this really nice video with the natural killer cell in action. It's only about 30 seconds, but it shows you exactly the formation or the interaction with the natural killer cell with a cancer cell. And as their interaction increases, you start to see how the cancer cell starts to implode and get smaller and smaller until it completely disappears. Another example of our innate defense system is going to be the induction of inflammation. Now, inflammation is usually seen as soon as there is an injury site, and the main benefits of it is to, and as you can see it right here at the bottom of your PowerPoint, it's going to help you prevent spread of the damaging agents. So we're going to try to localize the pathogens that have induced this injury all in one location, not have them spread out. We're going to actually start disposing of them and cleaning them out as well. We're going to set the stage for repair. And then more importantly, it's going to allow us to alert our immune system on a larger scale. And that also includes getting the adaptive immune system, so the one that's very specific with your T cells and your B cells, to come over to the site of injury. There are 4 cardinal signs of acute inflammation. So these are the signs that will always make an appearance. The first one is what they like to call redness. So if you hurt yourself, what we see happening is that particular area, when it induces inflammation, you're going to notice that it has a redness to it, and that is due to the fact that whenever you have inflammation, we're going to increase the amount of red blood cells that come to that location, partly because we're going to use the red blood cells to increase our oxygen drop-off and pick up our carbon dioxide, so we can kind of speed that up in that particular injured site. The next thing is that because you have so many cells coming over to the site of injury, you're going to notice that there is the presence of heat, that all these cells will contribute to that particular area as well. And there will be swelling. Now, the swelling can be caused by many different things. So we could say the swelling can be done due to the fact that you have more cells in that location. But the main culprit of the swelling is that what we see happening is that we're going to decrease the venous flow and increase the lymphatic flow. So we're going to make it slow down in the veins, and then we're going to increase the lymphocyte circulation. And the main reason why we're doing that is because, as you recall from your previous chapter, within the lymphatic vessels, we can encounter the lymph nodes, which will be able to filter out the fluid from the damaged area, so that if there's any type of pathogen, the lymphocytes will be able to detect it and, of course, alert the rest of your immune system. And last but not least, our fourth cardinal sign is going to be pain. And the pain is basically meant to make it less likely for your body to move that particular site. So it makes it easier for the repair and obviously the clustering of your white blood cells and any of your immune cells to occur. So redness, heat, swelling, and pain. Some books, and I did put this in our little PowerPoint, some books will mention impairment of function, but a lot of people will kind of piggy that off pain in the fact that it will actually just decrease movement. So we're trying to leave that area alone for the time of for the time that the healing process needs to take place. So for our purposes, we're only going to discuss the 4 that are listed in blue. Now, the stages of inflammation, meaning how do we actually induce this reaction where we can generate the redness, the heat, the trauma, and of course the pain that we were just chit-chatting about? Well, it turns out that there's going to be 4 steps that we're going to need to do to get our different white blood cells over to the site of injury. And along the way, we're going to rely on things like inflammatory chemicals, so chemicals that can basically signal that inflammation needs to be triggered or needs to be induced. And probably the most important inflammatory chemical is going to be histamine, but there's also some other like leukotrienes and bradykinin. But histamine is well studied, and we know that with histamine, it can cause massive amounts of vasodilation, meaning it's going to open up the lumen of our vascular system, of our blood vessels. In this case, we want to definitely focus on our arterial flow, because that's what's going to allow the red blood cells and the white blood cells to come over to the site of injury. And also we're going to see an increase of vascular permeability, which will make it easier for the white blood cells to hop out of the blood vessel into the tissue where the injury has occurred. So let's take a look at how these phagocytes are going to be immobilized over to the site of injury, and when they get there, what they're actually going to be doing. All right, so let's take a look at this illustration. So we have a piece of skin, and we can see right here that a nail has protruded through the epidermis and the dermis layer, and obviously that's going to go ahead and send off a defensive signal. So what we see happening is that the first thing is that once the inflammatory initiation is started, we see that we have our first step, which is going to be leukocytosis. Leukocytosis is going to follow the fact that primarily your neutrophils are going to hop into the blood from the bone marrow. Remember, your white blood cells do not like to stay within your bloodstream. They will basically only hop into the bloodstream when there is an injury that they have to fight off. So leukocytosis hints at the fact that these neutrophils in particular will go ahead and enter the bloodstream so that they can start flowing over to the site of injury. Now, in addition to neutrophils, there will be other white blood cells as well, but the neutrophils do play a primary role. Now, the next thing we're going to see is as they're in the blood vessels and they're moving around, when they get closer to the site of injury, we're going to see margination. Margination means that the white blood cells are going to start clinging to the capillary wall. And the reason they're clinging is because they're looking for an open spot so that they can do diaphysis. And diaphysis basically means that they're going to use that little open spot, and remember we have an increase in permeability, and they're going to use that to go ahead and flatten and squeeze themselves out of the blood vessels and into the tissue where the inflammation needs to occur. Once they're in the tissue, they will do their fourth step, which is utilizing chemotaxis. Taxis is movement based on a stimulus. So chemotaxis means that it's relying on those inflammatory chemicals that are being released. So remember our little histamine that we were just talking about? It's basically following the signal that these chemicals are releasing so that it can locate where the injury has occurred, and it can go ahead and start inducing inflammation. So we have leukocytosis, which means it hops into the blood vessel. Margination, it starts clinging onto the wall of the capillary. Diaphysis, which means it actually moves from the blood vessel into the injured tissue site. And then last but not least, it follows chemotaxis, which are the inflammatory chemicals, over to the actual site of injury. And then there we can go ahead and initiate inflammation. Now, your antimicrobial proteins, which also fall in your innate defense system, are fantastic at actually enhancing our immune response. And they can do it many different ways, and there are hundreds of antimicrobial proteins to discuss. So by all means, I have to put my little disclaimer that we are going to concentrate on the most important ones, which are going to be our interferons and our complement proteins. And even within those two, we're not going to be able to discuss all the details that they'll do. So we're going to look at what I like to call the big picture. But obviously, if you want to know more details, definitely send me an email, and I'll be more than happy to expand on my examples and whatnot. But either way, when you hear about the antimicrobial proteins, their main method is that they can either attack the microorganism directly, or what they'll do is they'll try to hinder their reproduction cycle so they can keep the bacterial or viral count at a lower number, making it easier for your immune system to control or come over to the site of injury. The most important antimicrobial proteins, aka the ones that we're going to discuss, are going to be interferons and our complement proteins. Now, interferons are a group of protein, or I should say a family of protein, that is released by cells when they become infected by viruses. And interferons are almost like an SOS signal, a little 911 signal. They will be secreted from the infected cells, and they will move over to healthy cells that are nearby, so neighboring cells, and they'll almost send off a warning signal so that those cells can go ahead and start producing antiviral proteins or any type of defense system that will make it harder for the virus to spread and enter into the healthy neighboring cells. So here's an example of our interferons at work. So we have two cells. This cell right here on the left-hand side is going to be our infected cell, and this one is going to be our quote-unquote healthy cell. So host cell 1 is infected, and host cell 2 has not been infected yet. So what we see happening is, let's take a look at the top of the illustration. We have our virus that's going to go ahead and enter into host cell number 1. And once that happens, it's obviously going to make contact with the DNA, because remember, viruses need to get access to your host genome so that they can replicate themselves. And as part of what we see happening is that, yes, the virus is going to replicate itself, but at the same time, the DNA that becomes infected will go ahead and induce transcription and translation and start production on these interferon proteins. These interferons are then released by host cell 1 and will travel over to neighboring cells, so all the cells that are nearby, and will allow the cell to process antiviral mRNA, and at the end be able to produce different antiviral proteins that will either block the virus from entering into the cell, or it will inhibit the viral reproduction. Either way, it will give the neighboring cells, in our example, host cell number 2, it will give them a heads up, a warning that the viral particles are out there. So interferons are proteins that are secreted by cells that have been infected by viruses so that they can send out a warning signal to their neighboring cells. Your complement system has been studied extensively. It is consistent of about 20 different blood proteins, and these are different segments of these proteins that kind of circulate around the blood. And what we see happening is that they have many different functions that they can do. For our purposes of our discussion, we're going to limit our complement system to the fact that they can enhance the inflammation response that the innate system is able to produce. And there's also examples where the complement proteins can actually be used to destroy the bacteria directly. They are a fantastic way of linking both the innate and the adaptive defense system. And oftentimes they will also increase the interaction between the foreign antigen and our antibody production. Now, I said a lot and all that, but what I want you to know for exam purposes is that whenever you see the complement, I want you to realize that it's a group of proteins, and for our purposes, its function is going to be that it's going to activate, it's going to help us enhance our inflammation response. Okay, so that's the function that we're going to limit our complement system to for exam purposes. But just, my little disclaimer, it does a lot more than that. And then our last one for our second line of defense, our innate system, is going to be the production of a fever. Now, fever by definition means that you have an abnormally high body temperature, and it turns out that that's done by the fact that your body will start releasing this chemical messenger called a pyrogen, and the pyrogen will travel over to the hypothalamus in the brain, and that is what sets your body temperature, and it will allow the hypothalamus to go ahead and increase your body temperature, thereby inducing a fever. Now, a fever can be a very good thing, and it can also be a bad thing. A controlled fever is going to allow the enzymes and proteins in our immune system to speed up, allowing for a faster response to occur. We also see that a slight increase in body temperature often makes it too hot for the bacteria and the viruses to replicate. So it's going to allow us to limit our load of foreign antigen within the body. We also see that it will cause the liver and the spleen to hold on to iron and zinc, which a lot of these microorganisms require for their replication cycle. And as you can see right here in the bottom, it will overall increase your metabolic rate, which makes it easier for your body to repair itself. Now, that's all in a controlled fever. If you have an uncontrolled fever, meaning that you start to spike about 103 or above, it really depends on your age and your body type and whatnot. Whenever you have an uncontrolled fever, it can be very dangerous because it creates so much heat within the body that our enzymes start to unfold and denature, and it can literally cause cell and tissue and organ death. So it can be obviously detrimental to the system. So a controlled fever is a very good thing. An uncontrolled fever can be a very dangerous entity. Now, a lot of times, especially when we're face-to-face and we discuss fevers, a lot of people will say to me, well, when I have a fever, I go and I get some ibuprofen, right? Because nobody likes the feeling of a fever. So we take ibuprofen or any type of antiviral medication, anti-fever medication. And what that does is it basically breaks the signal between the pyrogenes and the hypothalamus, and the hypothalamus will restore your body temperature to close to homeostasis, which is 37 degrees Celsius or 98.6 degrees Fahrenheit. When you do that, your immune system is still active. However, it's active at a lower rate. So we tend to see that you'll still obviously will heal, but it might take a little bit longer because you don't have that advantage of your increased metabolic rate and our increased response and the sequestering of the iron and the zinc and the inhibiting of the viral and bacterial reproduction cycle. So that's where that saying comes from, that if you have a fever, you should just sweat it out, because a short amount of discomfort will prove beneficial at the end, because it will often shorten your healing period. All right, everybody, I am going to stop part 1 right over here, and then I will see you for part number 2, where we will go ahead and continue to explore our immune system. But now we're going to go ahead and start taking a look at our specific immune system. So the one that we get exposure to, and the one that will develop those specific T cells and B cells. We shall talk soon.